Okay. All right. Yeah. So a little bit outline. First, give you some introduction into single molecule sensing AFM techniques, which is molecular recognition for spectroscopy recognition imaging. And then the requirements. So we'll focus more on how we prepare the tips for it. So most of the tips are made of silicon, and so I will go how we can functionalize the tips with small ligands for a simple test system, thiolated ligands, and then chemical coupling via amine residues, site-specific coupling, and photocoupling. And some of the tips are also made of gold. And there's actually I think mainly because of functionalization of gold is also a good pathway. I will also go into that, and then also how we can quantify the functionalization of the AFM tips. Okay, so that well, molecular recognition for spectroscopy. I already mentioned you can also measure unfolding forces of proteins that you just push into the protein and lift the tip and watch how the protein unfolds by force. But what I would like to tackle is more the measuring between molecules. Yeah? So between, let's say, a ligand on the tip and the receptor on the surface. So this is more what we are doing. And you can see the sketch here. So what we have to do is we have to functionalize the tip. Yeah? So we have to bind the ligand to the AFM tip. And we do this through heterobifunctional linker, which is a polyethylene glycol molecule. And the idea is that we want to provide the ligand that then binds to a receptor on the surface with sufficient motional freedom, yeah? So that there's no constraints of binding. Because if you would just attach it, adsorb it to the surface, it depends on the orientation and whatever. And we want to kind of also simulate the situation in solution a little bit. And this is why we are using this cross-linker. And then, of course, the receptor has to be bound to a surface. That can actually also done in the very same way as we functionalize the tips. Yeah? So you can also use the polyethylene glycol linker. It can also be a self-assembled monolayer, for instance. Yeah? Or what we are doing a lot, we are doing directly measurements on cells. Yeah? So we are doing force spectroscopy on cells. Yeah? If we want to use cellular receptors, this is actually the most convenient way. And you actually may think that this might be much more difficult with the cell. And this is what we also thought, but it is not. Yeah? Because cells are made to be anti-adhesive. Yeah? And surfaces sometimes are adhesive and there's some problems, but cells is a very natural environment. So it's actually, the mechanics of course is different, but it's very convenient to directly measure on cells. And I will in later lectures I will show you several examples where we're doing measurements directly on cells.
Cells also kind of clean in a way because they self-regulate their surface. Normal surfaces always quite dirty. Normal surfaces, you think about, you have to really clean it and make sure that the molecule of interest is there. But cells probably will have a self-cleaning mechanism.
Yeah, exactly. Yeah. So it is a very natural. Exactly. Yeah. And otherwise, I mean, cleanness is of course an issue. Yeah. Also for the tips. Yeah. But for the tips, you have to do it anyway. I mean, you can actually also bind cells to a tip, yeah, or viruses or bacteria. That's also what you can do. But you're completely right. I mean, the surface. You have to clean the surface, and then. It is quite tricky to really do it in a way that you don't have adhesion. And as you also, we started in '93 with the first force spectroscopy measurements, and the first publication was in early '96. So it took us quite a lot of time to figure out the proper surface chemistry for doing that. That is certainly a hurdle. Okay. And this is how it's measured. Yeah. So basically you have your receptors on the surface and your ligand on the tip. And then the first thing what you do is you approach the tip to the surface. Okay. And you measure, of course, the deflection of the cantilever, and the deflection of the cantilever directly translates into force. I think you were talking about this also yesterday about calibration. So okay, let's assume we are now far away from the surface. Okay, so this what you see here is distance, and what you see here is force. Okay, so we approach from far away to the surface, and then of course in the beginning the deflection is zero because there's no interaction. Okay, so you approach to the surface, and then you touch the surface, and you keep moving further downwards, and then of course the cantilever bends up. Yeah, and this bending up is shown here. So the cantilever bends up, and then you withdraw the tip from the surface. Yeah, but you're still in contact, and first the cantilever relaxes more and more. Okay, and if there would be no interaction at all, it would go back the same way. Yeah, but now let's assume that that the ligand has bound to the receptor, and then of course, if it's bound, then you have a resistance, and if you withdraw further, the bending goes into the other direction. Okay, and this is what you show here. So it goes into the other direction, and the force increases, yeah, because the cantilever bends more and more, yeah, and then at a certain critical force, you break the bond, yeah, you break the bond, and the cantilever snaps back into the resting position, and this is, and this jump here is the force that is required to dissociate the ligand from the receptor, okay, and this is what you can measure. And what is actually also important is this shape here. This is a single molecule signature, and this is what we get because of the linker. Yeah, and that is also convenient. So it's not only to provide the ligand with motional freedom, but we can distinguish between. So if it would be tip surface adhesion, this would happen here. Yeah, okay, but because we are using the linker, it is in a certain distance, and this already allows us to distinguish between unspecific, we call it unspecific tip surface adhesion, and specific dissociation. Yeah, and so this is also why this link is important. And I also want to emphasize that we are using polyethylene glycol. Why polyethylene glycol? Because that is also known in bioscience. It makes non-adsorptive surfaces. Yeah? And this actually signature, it is basically purely entropic, yeah, because the polyethylene glycol in solution likes to be coiled, yeah, just because of entropic reasons, and if you stretch it, yeah, then you need force for it, yeah, but it's entropic, it's basically purely entropic, right? It's not okay. And then we call it unbinding event or dissociation event. So there's different nomenclatures. Some people also call it adhesion. We say adhesion to everything non-specific, and so unbinding or dissociation event. Yeah? And this is how we can measure. I mean, we go into more detail then in the next lecture. And the second technique, which is actually which is really from our lab now. Yeah. Yeah.
So the smaller peaks that we are seeing in the straight line.
Yeah.
So that is because of the continuous tapping, or it's because of the thermal motion of the cantilever?
So the thermal motion of the cantilever, that's what you see here. Okay.
Okay.
Yeah. This is maybe a little bit of unspecific tip surface adhesion. Yeah. And because maybe it's also not the most beautiful force curve, because often it really looks the same here. Because here you also see a little bit of soft thing here. Yeah. So that's it's yeah, but so this is maybe because of the surface coating or tip coating a little bit that it's non-ideal, and this might be a little bit, they're not completely overlapping. Maybe it's also, it can also be hydrodynamics. Yeah? So if trace and retrace are shifted in force spectroscopy, that's a sign of hydrodynamics, because then if the tip goes down too fast, then it will already bend up because of the resistance in solution, and then the other way around. Yeah? So if you do too fast, then trace and retrace are a little bit shifted.
Okay. Yeah. Thank you.
I wanted to know that what information can we get if that snapping back happens at a larger distance? Let's say here it's happening at 40, but what can we say if it happens at 60? What does it say?
It is the stretching distance of the system in principle. So if you use a longer linker, then it will happen further apart.
Happen at a,
Yeah,
okay.
yeah. And if you do it on a cell,
Hmm.
this might go to 300 nanometers
Oh.
because you're stretching the cell.
Hmm.
The cell is softer
Softer.
than the spring constant,
Mhm.
and then it's you will see then in the later courses then it's 300 nanometers. Also, we are using the same
Yeah,
linker.
yeah, yeah.
So it's basically it's the elasticity of the
Mhm.
whole system.
Okay,
That's
okay.
reflects the distance.
Thanks.
Okay, and we are also using this deep chemistry for
Hi.
track imaging. There will be a
I have a question.
sure.
So, how do you find the position of the receptor?
The position on the receptor.
Yeah, on the substrate.
Okay, so basically, usually we are fishing in the dark.
Okay.
Yeah. So we have also a kind of high concentration of receptors. Yeah. This depends on what it is. If it's isolated, you can have a high concentration. Yeah. And on cells, you also have a certain density. But I mean, actually, if we combine now this technique, then we can also depict molecules. Yeah. Because with this technique, we are localizing receptors on the surface. Yeah. And the way we are doing it is that we are oscillating the cantilever that has a ligand, yeah, and we are scanning at the same time. And then in tapping mode, you're using the whole amplitude. We are separating the oscillation with the downstrokes and the upstrokes in this case, yeah. And you can imagine that if there's a protein, it will influence the downstroke, yeah, because it just cannot go that deep anymore. Yeah? So we can construct a topography image from the downstrokes, and on the other hand, if during scanning the ligand binds to the receptor, there's a physical contact, and this will influence the upstroke. Yeah? So what we can do here is we can, when we separate the downstrokes from the upstroke, we get two images at the same time. The topography image, which it's just like a normal topography image, and what we call recognition image. This is localizes where a receptor is. Yeah? And what we also did is, if you have a sparsely coated surface, that we first record where the receptors are with track imaging, and then we can go to this position and do force spectroscopy. Yeah? So in that way we control it. But mostly when we do force spectroscopy, we are fishing in the dark because we have a high concentration of receptors.
And one more question. So the tip has peg on it, right? And ligand is binded to that. So how are you preventing that ligand from binding to the peg on the substrate?
How to prevent the binding of the peg onto the...
No. Peg is also there on the substrate, right?
Can be on the substrate, yeah.
So how are you preventing the ligand from binding to the peg of the substrate?
The ligand binds just specifically to the receptor, not to the peg.
Okay.
Yeah. And this is also why we are using PEG, because PEG is a very inert linker, yeah, and it's used in biotechnology also for surface detection of yeah. Yes, yeah, that is yeah, right, yeah. Because it's PEG prevents molecules from unspecifically absorbing to surfaces. That's why PEG is used. So the ligand would not bind to PEG. And we are using, I will come to that, we are actually using, here the ligand is bound to the end group of the PEG. Yeah? So this is a very specific covalent binding of the PEG. But I will come to that there's...
Thank you.
Sure. Okay. Yeah. And of course, how we do the surface chemistry, it's not that just one ligand. What you see here is by the way, an antibody is bound to the tip. It's actually many of them, but the surface concentration is rather low, so that in average only one of the ligands on the tip has access to the surface. Okay, the others are on the side. So we can adjust the number of the ligands, and we do it such that the surface density is very low, that we can do single molecule force spectroscopy, and we bind the ligand covalently to the PEG linker, yeah, mostly covalently. And of course, this PEG has this nice elastic behavior, yeah, it's entropic chain, and important is the freedom of the movement around the tip, and also it doesn't induce any unspecific interaction between tip and surface. So this is how we use PEG. So we then can use different surfaces. So we are actually always using silicon or silicon nitride, which has the same chemistry. And of course, silicon tips are very good tip because they have a small tip radius. We can inactivate it. I mean, the protocols are rather time consuming. And gold has somehow an easier chemistry. It's faster, but the gold coating doesn't make the tip better. Yeah, that's also because it's also gold. I mean, should be inert, but if it's oxidized, then it's also a little bit hydrophobic. So we are not using gold tips, but you can also do that. Okay, so what is now the chemistry? So silicon tips. I mean, first we are cleaning the tips in plasma or ozone treatment. And then what is important on the silicon surface is the silanol groups. And they allow coupling via silanization. These are kind of standard protocols. Yeah, they are often used in biotechnology. So APTES, for instance, aminopropyl trioxysilane, or this AFS substance that's water soluble, or ethanolamine. We actually started out with ethanolamine, but now we are doing APTES. But we are doing APTES coating in the gas phase, yeah, because that is also important. If you do it in the water phase, it can polymerize. Yeah? So it is important to have very dry conditions, so desiccator, gas phase, salinization, that turned out for us to be the best method. Yeah? But clean tips are definitely important. And what we then, so basically we amine functionalize silicon surfaces with that. Yeah? So we have in a certain density, we have then amines on the tip surface for further tip conjugation. So first step is amino functionalization, and then we couple the crosslinker to the amines on the tip surface, and subsequently the ligands. And this is kind of 2-3 step procedures. And okay, so this is the chemistry for so the tip. There's now amines on the tip, and then our cross linkers actually carry all on this end a N-hydroxysuccinimide ester. Yeah? And this NHS ester binds covalently to amines. Yeah, that is also a standard amine chemistry, which is often used. And on the other end, we have a biotin, for instance. Yeah, that is then very simple. This is just 2 steps, and with biotin-related tips, we can detect avidin or streptavidin on the surface. Yeah? But we also have, here we have variable ends. Yeah? So here it's always a NHS ester, but here we have variable ends. And for instance, so the crosslinkers are heterobifunctional. Yeah? So they have 2 different functionalities on the 2 ends. One end is always succinimide ester, and the other one is variable. And for instance, we can couple thiols. So we have a maleimide linker where we can couple thiols. Yeah? So this is, we have all types of variable groups here. Or we have an aldehyde group. Yeah? That's also NHS. So that's the next example of the linker with an aldehyde group. And with the aldehyde group, we can couple to amines, for instance, to lysines of a protein. Yeah? So we can couple, every protein has at least an amine terminus, but often also amines, and we can couple proteins through lysines when we use a PEG aldehyde linker. Okay? Then, so the aldehyde linker has one disadvantage. As it can couple to amines, it could, for instance, also couple to amines on the tip surface and form a loop. Yeah? And this is what we don't want. This is why we are using acetal linkers, because acetal linkers have to be activated, yeah? When they bind in citric acid. And so that's why we saturate all amines on the tip first with the linker, and then we activate the acetal linker, and then we can also bind through lysines. Yeah? But it cannot couple back to the tip surface. One disadvantage of coupling lysines is you don't know which lysines you bind. Yeah? I mean, if you talk about antibodies, antibody has a lot of lysines in the Fc, and this is how we want to bind. We want to bind the antibody more here, because the antibody binds here to the antigens. Yeah, on the very antibodies are Y-shaped molecules, yeah, with two FAP arms, one Fc arm, and the end of the FAP arms bind to the antigen. And then we have another method that we can. Antibodies also are glycosylated here at the hinge region, and we can also use these sugars to bind it to the tip. And for this, we have to activate the sugars by this treatment, but then we can specifically bind the antibodies there and ensure that the antigens are on the other side. Okay? Then I think you probably all of you know hexahistidines. Yeah? Hexahistidines are often used on proteins, so they are genetically modified, the proteins, for a hexahistidine tag, and they are also used for purification of proteins, yeah, on NTA columns. Yeah? So hexahistidines bind specific, not covalently, but very strongly to NTA. Yeah? And this is why we also have NTA tips, where we can bind hexahistidine, so proteins that carry a hexahistidine tag directly to the PEG linker. Yeah? And it's not a covalent binding, but it's a very strong binding. Yeah? And of course, then you will see it then also later in applications. Then of course we are using the hexahistidine tags are opposite to the binding site of the ligands, and this is then also a very site-directed binding. Yeah? Because site-directed binding is important, because the forces that you measure depend on the direction of the force. Yeah? So site-directed binding is very good. Then there's one thing that is binds a little bit also covalent, but not site-directed. This is benzophenone. This binds more to hydrophobic regions of a protein. Yeah? So, for instance, you can use it for any organic molecule, proteins, nucleic acids, but they bind more to kind of CH bonds, but a little bit unspecific, but covalent. Yeah? So, but this is also something what we already used. But actually, what we are mostly using is amines we use mostly with drugs or so. Yeah? Then for the antibodies, we are using the oligosaccharides, and we also like to really use the NTA linkers and apply it to to proteins that have a hexahistidine tag. That's what we also do very often. Oh, yeah. So just to give you a summary, what we have, that is biotin, yeah, for binding to avidin. Actually, what also can be done is that you bind then avidin to a tip and bind a biotinulated protein to the avidin on the tip. Yeah, that is also a possibility. That is also strong enough, kind of. It's not covalent strong enough. And covalent linkers is aldehyde for binding to amines like lysines. But we are preferring the acetal linker here because it doesn't form loop. Benzophenone binds to hydrophobic regions. NTA binds to hexahistidine groups, and then yeah, I think this is the acetal. No, this is thiol. If we use malamide, we can bind it to thiols. Yeah, so because you can also genetically engineer proteins and put a thiol in, cysteine in, and then it's also site-directed binding. Okay. Yeah. No, it's not one link. It is actually. Let me go to this sketch here. It is more than one linker. Yeah. So the distance of the ligands is determined by the first step of the chemistry, which is the APTES functionalization. Yeah. And what we get here is a kind of distance of 10 nanometers. Yeah, that is the number density of the APTES sites. Yeah, but it's dilute enough that only one ligand here on the very tip end has access to the receptors. Ah. Okay. Yeah.
The density of these ligands you have kept like few at the tip so that only one ligand bind to the receptor at a time, but is it like independent of the receptors that is present in the biological sample? What if the density is high there, and won't it change the probability of these ligands and receptor binding?
Yeah.
So that it might make more than one, like at a time more than one ligand is binding to the receptor. Then
it is somehow also, it also works at a high density of receptors because this molecule cannot reach the surface. Yeah? However, for instance, if you have a cell surface, of course the tip indents a little bit, and it's not 100% strict. So sometimes we have a second molecule on the tip, but then you see it. Yeah? Because then it has two disruptors. So what is actually happening? You see it in the force spectroscopy curves. It's not 100% strict, but it works very well. Then if we have a tip that shows too much binding, we just put it away. Yeah, but we can use like 60% of the tips are very nice for just one ligand.
okay, thank you.
Okay, and as already mentioned, we can also use gold chemistry. And one time we used a tetraeder with aptamers, which was thiolated, and we got very nice force curves with that. So it also works. Yeah, but it's another group that does more. But you can also use it. You can form like tetraeder. Now you know all this DNA origami stuff that has thiols on, and then they bind. Actually, in this case, they bound very defined to a gold tip. But this is just actually one publication that we did. And also, there's also a possibility if you really want to have one ligand. That you can pick up the ligand from the surface. This is what we tried with the tetraeder. But there's also a publication from a friend of us who actually showed then also with fluorescence that he could pick up a single ligand molecule. But that is, of course, a lot of effort to do it in a way with each tip. And we are fine with our chemistry, how we do that we have single molecule. Yeah, and the question is, how can you quantify how many you have on the tip? I mean, you could do it with electron microscopy, but you know, one protein is not easily visible, so you can maybe put a bead on to these molecules. There you can do it, or you do an assay which is horseradish peroxidase. I think you probably also know it, where you can quantify how many molecules are so just by fluorescence. Yeah, so you bind the fluorescence signal is proportional to the number of horseradish peroxidase molecules. There's a certain assay how you can transfer it that it gives the fluorescent signal, and there you can define the density. You can measure the macroscopic density, and with our chemistry, it's something like every 10 nanometers molecule. That's the average density, and that works very well. Okay, or you can and of course, on the surface, you can also use track imaging to quantify the number of molecules, but of course not on the tip surface. There you just use in parallel a silicon nitride surface, and can also do this quantification there. Okay. Yeah, that is a little bit. I think I don't go into this. This is a little bit specialized. I just want to emphasize that as I'm a physicist, I could not do this development, but it was a long-lasting collaboration with Hermann Gruber, we call him our chief chemist, and he retired almost 10 years ago. And in the beginning, I was really a little bit worried because he synthesized all the linkers, and he also kind of sold it in a way. He didn't really become rich of that, or his group didn't become rich of that. But now everything is commercially available. So if you can just, there's several companies like Creative Packworks, for instance, or also Polypure in Norway, and you can get now all crosslinkers commercially available. Okay, all right. Then thank you for this part. Of course, if you have a different.
Single molecule recognition imaging. So you have like the upper part of the amplitude and the lower part of the amplitude. I think there will be like two feedback system for two different imaging. One is for topography image, another one for recognition image.
So it's the same frequency. When you just here, we are separating.
Not frequency feedback system.
Two feedback system. No, there's just one feedback system. We just put feedback on the topography.
Okay. Oh, so then how we get the recognition image? Okay, it's not like the tapping mode. We set a amplitude and fix the amplitude and get the topography image.
I mean, you can also server on the end. It also somehow works. There will be an own electron tracking machine where I can show you how the feedbacks. Yeah. But the cleanest method is to put this signal into the feedback loop.
Yeah.
And then basically also the feedback loop holds the signal constant. That's it follows the topography. And on top of that, we detect these peaks, which uses the recognition.
Yeah, the problem is when it goes in the like surface, once it binds, the feedback may not work like properly because it binds to a particular molecule that we are imaging in the recognition imaging. So that may create problem for the topography image. So what about that?
I will also present it in detail. You're right. What could be a problem is the crossing of the two signals.
Yeah.
And in that case, it is very important to work with the low Q factor. Yeah. So basically, that after a half oscillation, because these two signals are separated by half oscillation, right?
Okay.
And because it's up and down, yeah. And so if you have a Q factor of 1, then basically the information has been almost forgotten. But you're right, if you have a high Q factor, then these two signals influence each other. So you need a high density, in other words. Otherwise, you carry the information from here into this channel, and of course that doesn't work.
Hi Peter, here, here.
Hello.
So you somewhere mentioned about the angle at which we pull. That is also important. So how do we incorporate that in the instrumentation? Is just piezo that you use to pull at different angles, or how is it done?
Yeah, that is maybe also the advantage of the linker that it adjusts, to a kind of neutral position. I mean, it's not like so the influence is very small. If you are saying like you are 1 nanometer here or 1 nanometer here, there's not much. But where there's influence, if you and this has been shown in a work with Abhi. If you enter the Abhi molecules on different positions, then you really get a different dissociation angle, even if you use a linker. And that is dependent on the force value. I mean, force is a vector, right? And so if you look like atomistically into the binding pocket, then one can also show it with MBS. Then there's a difference, if you're in the binding pocket, and then it's different if you do like this or if you do like that. But it's more how you anchor the protein rather than being 1 nanometer there or 1 nanometer there. That doesn't matter.
Okay. Yeah. Thank you.
Sir. Sir.
Yeah.
Yes. Sir, I
Yeah. Can you speak a little bit louder, please? Or maybe sit down. Okay.
modifications of the tip. Uh, yes. Or like means how is it actually?
Sorry.
Okay, sir.
Sorry.
Sir, so like for this chemical modifications, can this be done on any tip, or are there any specialized tips that are required for this? And also, are there any chemical modification kits that are available by which I can probably want to link my protein of interest via the cysteines or the lysines?
Yeah. I mean, of course, the surface has to be chemically addressable, right? And this is silicon is the usual agent, and silicon chemistry was developed much before AFM. So the desalinization for biosensors, silicon surfaces are heavily used. The same is for cold surfaces. Also, cold chemistry for biosensing is used with the self-assembled monolayer, and this is also the reason why people cold coated AFM tips, because the chemistry is easy. Then there is carbon tips. This electron beam deposited tips, they are made of carbon. Carbon is actually really inert.
Okay.
And it's difficult to functionalize. However, we did it. So with our chemistry group, it is a very strong oxidation what you have to do with carbon, but then you can also functionalize carbon. I mean, these are the tips. that are commonly used, and you can modify them. I don't know, for instance, tungsten tips are more used by STM. I don't know if you can functionalize tungsten. I think it's also chemically rather inert. But I would say, like for gold, silicon standard recipes are available. If you want to functionalize EPD tips, then contact us. We can do it. That's EPD
Okay.
published yet.
Okay, sir. Sir,
And okay, this was the first thing, and the second was then if you're using different. Uh,
like suppose I'm trying to link my protein of interest via the cysteines or the lysines. Uh, then like are there any kits available to like attach a malamide linker to the
yeah, this is given by. You mean the protocol for doing the TIT
yes.
functionalization? Uh, yes, we publish them, for instance.
Okay.
And sometimes I think if you are also looking into the literature, I'm not sure how, for instance, creative networks, they also provide protocols.
Okay.
But usually, so we actually published all protocols, so it should.
Sir, and like are these tips just for one time use or they can be
No.
used for
You can
multiple times?
use them for. It is more actually dependent on the stability of your ligand. Yeah? But honestly, it was one time that we then recognized that we should maybe do one more experiment, and then we looked into the fridge and we found a tip that was 6 months old, with an antibody, and it's still.
Okay. And so just one more thing. So suppose I'm trying to see how my protein of interest it binds to the substrate. But say there are like multiple cysteines or multiple lysine residues that are distributed all along the protein. So like I cannot specify that like that malamide linker will attach to a specific cysteine. So like suppose that malamide linker attaches to some cysteine that is near the active site and it alters the active site conformation. How do I take care of that?
There is exactly the problem of the non-site directed pathway. So that both with lysines and cysteines, there because proteins with many lysines. And this is why we are preferring the
Hexahistidine.
that's site directed. Same is with the cysteines. We are using amine sometimes for very small drug molecules, but then these drug molecules have really only one amine on. Yeah? There we are also using amine chemistry. Yeah, one thing I also would like to mention, we also have techniques with an alkyne group, so we can also use the click chemistry, which is also something. Yeah. So in principle, you could use, there's also other very stable packs available. I mean, there's even, how are they called, SnapTech, I think. Yeah. I think SnapTech is even more stable than the existing pack. Yeah. This is also something that we never did that, but other people also use SnapTechs. But it's really, I really would recommend to do a site-directed coupling, that you know which group it is coupled and get up to state, because you might have a lysine in the binding pocket or system. And also you get variety in that. And people are actually in the literature also, and I can see it, because you get a high variation of the forces, because one protein is coupled with this lysine and the other one with that, and you get different forces, and then you get really a mess of data. But yeah.
Hi sir, during this recognition, some falling processes happen or not?
Some
Falling because we using bio-recognized compound, some ligand and all. So just for example, we are working on biosensor. Then we're doing some sensing. That's falling process is very high. So this is a big problem for biological sample. So here also we can recognize tip using biological ligands like that. So during this process, some falling process is there.
We don't really see a fouling process. I mean, this is not working? My, okay. Okay. The battery. Okay, okay. I'll give it to you. I mean, we didn't really recognize fouling process, but maybe we don't use them. So we like to use fresh ones. Yeah. Of course, what also can happen is that you pick up something. This would also happen with the non-functional tips. So using them maybe for a week or so, then we prefer to use it weekly. But so we didn't really specifically, but maybe just because we are changing it quite frequently. Okay, yeah. Maybe wait for the mic.
Yeah. So I was wondering, while doing the force measurements, have you recorded the lateral signal and does it give any important information? Is it helpful?
Which signal there?
The lateral signal of the photodiode.
Lateral signal,
Yeah,
torsional signal.
yeah, torsional signal.
Not really. I mean, first, actually, we wanted to do localization of receptors on surfaces using the friction signal. And, but of course, the torsional spring constant is at least, I think, 100 times higher than the vertical one. Yeah? And so it's not very force sensitive. We are not, for force spectroscopy, we are not recording it at all. And I think very initially, we tried to use it maybe during scanning, but then it was not sensitive enough. And I think that's the main problem. But I think in the meantime, there's publication out where they are also using the lateral signal for so for recognizing receptor sites on the surface. I saw it one time. I didn't really go into that, but that's the problem of the torsional spring constant, that it's not very sensitive, and also not very much used in biology. In tribology, yes, of course, but in biology, there's not a lot of literature on it.
Sir, just one final question. You said photo coupling through benzophenone. So there the light, the photo, what does it do to the benzophenone group? Like, does it create a conformation change? To, like, how does light play a role in that?
Oh, I think I don't think, because I think once I would imagine, I'm not a chemist, but I think once the benzophenone is attached to the protein, it probably cannot do these conformational changes anymore. This is what I would speculate.
Okay, so light, what does it play a role? Like photo light is.
In general, or
In general, for the entire process, like what does it play a role?
you mean now the laser that is focused on the AFM tip, or?
No. Because the process is called photocoupling. So does it?
I see what you mean. Okay. Let's see, where do we have it? Coupling, what do we have there? Photocoupling. Actually, I don't have now the exact conditions in mind for that. So I don't know if it has to be done with some light or not. It says
Light of
360
yeah.
nanometer.
Okay. Thank you.
Light is.
we didn't use that very often. Yeah. Okay. So apparently we need 360 nanometer that the coupling works. Yeah.
Okay.
Yeah.
So at that wavelength, the reaction occurs.
That it seems that this induces the reaction that it can bind to a protein, or so you need 360 nanometers. Yeah. Okay.
And so one question, this gas phase selenization you're mentioning, which makes it like more even, the selenization. So after gas phase selenization, we have to keep it in inert argon atmospheres.
Yeah, that's right. Yeah.
So one thing, like when it is for a purpose, we need to take it out of the argon atmosphere, and like let's say for 2 hours we have to keep it in the outside atmosphere, and we know that if the outside atmosphere is oxidative, so how damaging can it be for the aptus layer?
Well, we actually put it right from the desiccator into a solvent for the further fractionalization.
I know, but since I've followed one of the protocols, we actually for a purpose we need it inside the outside atmosphere for, let's say, half an hour or 1 hour. So how secure would the aptus layer be after that? And after that, like immediately you have to like put it inside the liquid, but within that half-hour time period, how secure like aptus will like?
Honestly, I don't know. We never tried that. So we are just trying to really store it in argon in the desiccator and then immediately put it into solvent for the next step. And we didn't, vary that. We are just trying to be careful with that.
Thank you.
Yeah, thank you.
Sir, so when we're talking about like ligand binding with receptors, like there is also receptor dimerization, right? Like, so how do we actually study that?
Dimerization, yeah. I mean, we didn't really go into that. We had kind of had some plans to, and actually we also did it to produce branched crosslinkers. Because for some membrane proteins like transporters, dimerization or even quatermers play a role. Yeah, and we went into the synthesis of the linkers, of branch cross-linkers, and where we could maybe see that if two are binding or so, but somehow somehow the project then stopped and we went into something different. But I think what you really would have to do is to study it with branch cross-linkers and see how they are dimerized. We just thought then there was a colleague who did fluorescence microscopy and I mean this transport that they had a GFP attached, and he made then a detailed fluorescence assay when they dimerized, and he could basically measure the aggregate state. And I think then we thought, yeah, maybe it's easier to use fluorescence microscopy with that instead of for spectroscopy with branch cross-linkers.
Okay, sir. Thank you.
Thank you.